Here we have a Chitosan hydrogel, which was fabricated using electrodeposition. Electrodeposition is a newer and promising method of manufacturing which can allow for many unique capabilities. So let's learn about what electrodeposition is and what it's all about. An electrolytic cell is the foundation of electrodeposition. Electrolytic cells typically contain three main components: a battery or power source, electrodes, and a solution. The electrodes will be labeled as either the anode or cathode depending on their connection to the positive or negative terminal of the battery. It is important to note that electrodes must be some type of conductive material to allow electricity to flow through it. Now, when looking at the flow of electrons, let's see what happens. These electrons start at the negative terminal, pass through the cathode, into the solution, back into the anode, and eventually into the positive terminal of the battery. However, electrons can't just flow through a solution, so let's take a closer look into what is actually happening. Now, in order to describe this movement of electrons, let's start with the cathode. In a solution, we will typically have some hydrogen ions. These hydrogen ions will interact with the cathode and electrons will be donated from the cathode to the hydrogen ions. This results in the formation of hydrogen. Then, at the anode, we can start with two water molecules. And when they interact with the anode, this will cause electrons to be moved into the anode, leaving hydrogen ions and oxygen. These two interactions are the basis of another important concept in electrodeposition, which is the pH gradient. These interactions cause a localized pH gradient around both of the electrodes with a low pH at the anode and a high pH at the cathode. Here we have our experimental setup to demonstrate the pH gradient. We have the anode on the left and the cathode on the right. These electrodes are made out of platinum foil. The electrodes are submerged into a solution of pH indicator, agar, and sodium chloride. If we press play, we will be able to see what happens when an electrical input is applied to this experimental setup. As we see, the solution is changing colors locally to each electrode. At the cathode, we can see a darker color, indicating a high pH value, while around the anode, we see a lighter color, signifying a lower pH value. Now is a great time to talk about the electric field. It is another concept that plays a crucial role in electrodeposition. So, if we start with two positively charged molecules and induce an electric field, we will see the molecules move in the direction of the cathode. This is important because the electric field can play a role in the movement of positively charged molecules away from the anode and towards the cathode. This movement can help explain some parts of electrodeposition. As we know, some molecules are big, some are small. However, it is not uncommon to see these larger molecules being made up of many smaller molecules. The larger molecules that are made up of the smaller ones are called a polymer, while each of the smaller molecules are referred to as monomers. It is easy to think of a monomer as one unit of a polymer. A few examples of commonly seen polymers are polyethylene glycol, known as PEG, which is a synthetic polymer, and its monomer is ethylene glycol. Another polymer we are familiar with is RNA, which is a biopolymer, with nucleic acids as the monomers. The specific molecule we will be talking about today is chitosan, which is a polymer made up of glucosamines as the monomers. Now, chitosan is unique in the fact that the amine group will change depending on the pH value, changing the properties of the molecule. As we can see, when chitosan is in a lower pH value, the amine group is protonated and it is soluble. However, when chitosan is in the presence of a higher pH value, the amine group is neutralized and the chitosan becomes insoluble. When many chitosan chains are present, the high pH value can lead to an interesting mechanism from chitosan called self-assembly, which we now have our colleague Ariane to tell us about this.
Chitosan is a linear polymer composed of sugar units linked by covalent bonds. These monomeric sugar units link to form large chains which tend to associate with each other by hydrogen bonds.
As we can see, we start with two chitosan chains, and if we zoom in, we can see that there are many small bonds formed between these chains into what is eventually a sheet. This is the basic concept behind chitosan self-assembly. What we see demonstrated here would be considered the perfect crystalline structure. It is important to note that that is not the exact structure that we receive from electrodeposition. Here, we can see a better representation of what an electrodeposition experiment would look like when using chitosan. As we can see, locally around the anode, the chitosan polymers have positive charges which will repel the chains from each other and prevent self-assembly of the gel. But, as expected, we can see the chitosan that has been neutralized on the cathode can self-assemble into a gel. This is how simple it could be to perform electrodeposition, which can be the future of additive manufacturing. Here is an example of how we can form a complex structure using electrodeposition. First, we start by turning on the electrical input. This will cause the normal gel to form as expected, which we will refer to as a segment layer. But if you turn off the electrical input, a denser layer will form, which is referred to as the boundary layer. If you keep oscillating the electrical input between on and off, you can form the complex structures on a micro scale within a gel. This is an example of what a gel made using oscillating structures looks like. This specific experiment was formed around a wire, causing the circular nature. As we can see with the scale, the uniquely structured layers are only micrometers long. This was discovered by one of our colleagues. Let's see what made them think of this.
Based on a report from France, we decided to try to deposit chitosan using a sequence of on-off inputs.
Now that we have seen some interesting ways to manipulate chitosan, we will now talk about the application it has in the real world. One known use for chitosan electrodeposition is Janus porous film. This film is constructed using two separate solutions, starting with a solution containing a low salt content, which is deposited to form a dense surface of the gel. Next, the solution is changed to a higher salt concentration, which will result in a porous surface to assemble. This certain film can be useful in the medical application of guided bone regeneration. The dense layer can provide support and prevent tissue regeneration, while the porous layer can promote osteogenesis and recruit osteogenic cells. This is how chitosan electrodeposition can be applied. Now, let's talk to one of the people who found out about this method.
Electrical fabrication is an attractive manufacturing technology for functional materials. Our group focuses on the development of biomedical materials, and using this technology, we can fabricate functional materials with an isotropic porous structure, and the special structure of the film is beneficial for guided bone regeneration.
Learning about electrodeposition can be a lot to take in, but there are a couple cool things that are important to take away. First, electrodeposition is seen to be simple, safe, and inexpensive. In the pictures, we can see a chitosan electrodeposition experiment being performed successfully using only a 9-volt battery, pencils, and alligator clips. This can show how easy and simple it really is to create a hydrogel using this method. The next important takeaway is the customizability of these materials. As we mentioned before, the complex structures such as this segmented gel can be created. Also, these electrodeposited gels allow other methods of customization, such as co-deposition, which can be seen here using green fluorescent beads on the same pencil deposition setup as shown on the left. It is important to think about the endless capabilities electrodeposition and hydrogels have in the manufacturing world. Here, we have one of the first people to ever deposit chitosan. Let's see their thoughts on what has come from it.
When we first discovered chitosan electrodeposition 20 years ago, we were trying to create a biocompatible interface to integrate biology into electronics. Now it is exciting to see chitosan electrodeposition emerging as a new method of additive manufacturing for a broad range of applications.